Hello and welcome to today's webinar, Powering Phase 4 Trials, Design Considerations for Post-Marketing Surveillance Studies, presented by Dotmatics nQuery. So the hope with today's webinar is to give you an overview of what Phase 4 trials are and the specific area of post-marketing surveillance focused on adverse events on safety reporting. When a drug has been approved after Phase 3, and now we want to establish safety in the real-world population and real-world patients, in this case via a formal study or clinical trial. Before we go any further, just a couple of frequently asked questions. First, is this webinar being recorded? Yes, and the recording alongside the slides will be sent to you after this webinar is complete, either later today or early tomorrow. The slides will also include a number of references, which I'll talk about throughout at the end, alongside links to doing a demo of the software. I'll talk about that at the end, and also an email address if you wish to get in contact after the webinar. If you want to ask a question during the webinar, there is a questions tab on the right-hand side, I believe, of your webinar software, and you can put a question in there. I will attempt to answer some of these at the end if time allows, but if time doesn't allow, then please be reassured that I will answer all questions via email after the webinar is complete. So please do use that option if you feel the need to. So as I said, today's webinar, we're trying to look at Phase 4 trials, post-marketing surveillance, the situation where a drug has been approved for clinical usage. It's now out there in the real world where you can use by real patients, but there is some reason why we want to do a study, in particular if we're doing a Phase 4 study, to try and establish most likely safety concerns that may exist if we're talking about a requirement from the regulator. But we'll talk about that later on. So my name's Ronan Fitzpatrick. I'm the head of statistics here at nQuery. I've been nQuery over a decade now, and we've done sample size determination on a wide variety of topics throughout these webinar series, both myself and my colleagues. But this is the first time I believe we're covering Phase 4 trials. I'm excited to get into an area which many of you may be less familiar with than with Phase 3 trials, with earlier stage trials, with preclinical research. So hopefully you get some really interesting information out of today's webinar. So in terms of what we're going to cover today, we're first going to cover post-marketing surveillance, which is really an ongoing process which is happening all the time with or without something called a Phase 4 trial occurring. And really the terms post-marketing surveillance and Phase 4 trials are often used interchangeably. I'll try to distinguish between them as we go along to reduce confusion. And then we'll talk about Phase 4 clinical trials or Phase 4 studies. Those kind of terms kind of mean similar things, but maybe not quite the same thing. I'll talk about that later on. And then in the third section, we'll talk about, okay, you are doing a Phase 4 study or clinical trial. What are the design considerations that are most important when trying to design the appropriate study for whatever your objective is, most likely trying to find like adverse events, whether rare or non-rare. And at the end, we'll have time for conclusions, and we'll have some discussion then if time allows. As I said, questions, please give them as you go along. Even if we don't get them to today, I will answer all of them via email, and I'll provide links and stuff, even though I do answer today in an email reply to ensure that you get the full insight that you were looking for. So today's webinar is presented by nQuery, the complete solution for optimizing your clinical trial design. So nQuery is a software that's focused on sample size determination, power analysis, adaptive design, simulations, etc. It covers these from preclinical research, animal studies, etc., lab studies, early phase designs, Phase 1, and in particular Phase 2 with Simon's Designs, NC Mod, etc. And for confirmatory trials. Like confirmatory trials is probably the big area where nQuery has traditionally been strongest, where sample size determination and adaptive designs are very common and are based on statistical criteria like power, statistical significance, etc. And we have to quite wide variety of scenarios as confirmatory. But as I said, today we're going to focus on this kind of post-marketing section, this Phase 4 section. We're going to talk about observational studies in particular. And this is something which you haven't covered in the webinar series before, so I'm excited to get into that today. But you can see nQuery is a very thorough software. So if you're looking to do clinical trial design related work, sample size work, adaptive designs, then nQuery is a very complete solution for doing so. And we're happy to talk about that further if you email us at info@statsolves.com. Just to mention, nQuery is a tiered product. So effectively, the bottom tier basic covers sample size and power calculations. Plus covers our Bayesian features like assurance or probability success calculations, or sample size for things like credible intervals. Pro covers our adaptive design features like group sequential design, promising zone designs, multi-arm, multi-stage designs, phase 2 adaptive designs, etc. An expert is focused on our simulation tools, particularly right now on milestone prediction for enrollment and event milestones, how long it takes to get to reach a milestone events, for example. And this is a tiered product, which is to say, if you buy the expert tier, you get all the previous tiers. Pro gives you plus and basic, etc. So hopefully it gives you an idea of what's in the different tiers. If you want more detail, there's a URL there you can see at the bottom. So the slides will be sent to you after this webinar is complete. You can use that URL to investigate further which tier is most suitable for your needs. So with that out of the way, let's get into today's webinar and the educational information that hopefully be of great interest to you. Starting with post-marketing surveillance. And I suppose, as I said, there's kind of oftentimes in the literature, when I was kind of going through different sources on this, a confusion between phase 4 and post-marketing surveillance. I'm going to talk about post-marketing surveillance first, even though some authors would argue that phase 4 is really the wider concept. And I'm going to do that because post-marketing surveillance is something which really happens regardless of whether you're doing a study or not. There are publicly available databases from regulatory agencies like the FDA and the EMA for drugs, biologics, for medical devices. And these are obviously getting information from doctors, from clinicians, from patients, from sponsors, from the regulator themselves, which are obviously then providing ongoing information, ongoing surveillance of what's occurring in the real world in terms of safety outcomes in particular. And that's obviously something we would expect to happen. It's something which does happen, and something which phase 4 trials are often a response to or an addendum to, if we're talking about a kind of a specific, different or like separate phase 4 trial compared to just the ongoing post-marketing surveillance. So obviously, post-marketing surveillance, as would be implied by the term, is where we're monitoring a therapeutic, a drug, a biologic therapeutic, or a medical device in the real world, in patients who are getting this therapeutic, this device, since it has been approved for clinical practice. So for a drug or biologic, it's passed the phase 3 trial assumptions. We've got 2 successful phase 3 trials, for example. The regulator, like the FDA, decided the drug was, suitable for usage in real-world patients due to, superior performance in terms of efficacy or other considerations. And, now we want to, now it's actually been given to real patients by real clinicians. We want to, continue to ensure that it's giving, that these therapeutic is operating as expected. And in particular, in a phase 3 trial, while being large, hundreds of thousands of subjects, they're quite controlled, right? You have inclusion-exclusion criteria, you have tight monitoring of what's happening at each of your sites, or at least hopefully you do. Hopefully you have that. And of course, that means that, compared to the messiness of the real world, where obviously, non-compliance is very common, between patients, the doctor or clinician maybe choosing between multiple potential therapeutics, and they may do that based on other like confounding factors. Obviously, this is for all patients, children, older people who may have been excluded from the clinical trial. We want to see what's happening in quote unquote the real world, in real world patients, and that's what post-marketing surveillance is designed to do. And of course, we're trying to cover a wide variety of different outcomes here, and some of them are more interest to regulators and the public interest. Some would be more interest to sponsors and in terms of their economic interest. Like the one we're going to focus on mostly today is safety, and in particular, like rare side effects, which may not have been found in a clinical trial because they're rare. You might need tens or thousands of people to find these. And so adverse events are really what we're going to talk about mostly today, but you may be interested in finding efficacy signals, whether that be, certain subpopulations have better efficacy or lower efficacy than other subpopulations. Maybe there's alternative indications which may be of interest for a therapeutic that are then found in the real world. So it wasn't via phase 4 or post-marketing surveillance trial. Technically, it was more done by the sponsor themselves during phase 3, if I recall correctly. But, an example would be the GLP-1 antagonists like Ozempic, were found for treatments for type 2 diabetes, but were found to be a successful treatment for obesity, or at least that's what the approval of Wegovy suggested. And of course, that's a case where an additional indication became available and formally approved. Obviously, off-label indications may be of interest as well. We're going to talk mostly about safety today, but just to say, like, post-marketing surveillance and phase 4 studies can include efficacy as consideration. And I say, like, in terms of safety, post-marketing surveillance is particularly important where there was an accelerated approval, where you may have used the surrogate endpoint. So, for example, you might have used progression-free survival rather than overall survival, or you might have used overall response rate rather than PFS or OS in say, oncology. You may have additional subpopulations, like the pediatric population is an obvious candidate, but maybe other minority populations may have been underrepresented during the clinical trial, and you may want to have specific post-market surveillance, a specific phase 4 trial, for example, to deal with what may be happening in that population. And medical devices, the entire approval process for medical devices is quite different from drugs and biologics. For example, at phase 3, it's pretty normal to only have one phase 3 trial rather than like either 2 phase 3 trials or a global phase 3 trial. And so that, and for many medical devices, they get approval based on being considered to be equivalent to existing safe devices. So medical devices is a whole other regulatory pathway, and so post-market surveillance is particularly important there, and the databases there tend to be particularly thorough because the level of stringency for a medical device getting the market is less than for typically for a drug or biologic, unless it is a high risk category type medical device. And so the main focus today, what we're going to talk about today in terms of like the post-market surveillance concept, is really where we're going to do a phase 4 trial, a phase 4 study, which is a focus on pharmacovigilance, which is really safety related surveillance effectively. And of course, we're kind of really doing, we'll talk about kind of doing pharmacoepidemiology, where we're not doing clinical trials anymore in a real sense, or more often we're not doing that. We're kind of doing epidemiology, and we'll take the epidemiological type studies like cohort designs and case control designs. And what we're going to kind of focus in here today a lot is on the type of designs and the type of suggestions made by the FDA in particular for when a phase 4 trial is being put forward as an addition, kind of an addendum to post-marketing surveillance in response to some specific either requirement or to some new information that became available. So phase 4 trials. Probably useful to have a decent overview of what the clinical trial process is like, so we can kind of get a context on phase 4 trials. So really, when we're talking about getting a drug or biologic or other therapeutic approved, as I said, medical devices broadly the same, but there are some differences. We basically have 5 main stages. There's preclinical. This is basically where we're investigating a wide variety of different potential drugs, molecules, biologic entities, proteins, etc. And we're obviously trying to find ones which we think will be effective based on animal models, based on in vitro studies, based on cell studies, etc. We're trying to find, obviously, those candidate therapeutics that we're going to bring forward into the clinical trial process and hopefully get approval, hopefully establish that the therapeutic is safe and effective and approvable for usage in real patients and to provide real benefit to patients in the real world. And so we start in phase 1, which is our first in human trials, often done in a population which doesn't have the disease of interest or the condition of interest, though sometimes they will. For example, oncology, sometimes they will. And in this case, we're usually just trying to find the acceptable range of doses for the drug of interest. And in oncology, this will often be trying to find the MTD. The assumption in oncology traditionally, especially with chemotherapies, radiotherapies, was that the higher the dose, the more effective it is. And therefore, we want to find the highest dose which has an acceptable toxicity profile. So the maximum maximum toxicity dose, tolerable dose here, is the MTD, and we basically want to basically find that so that at the subsequent phases we can just start there and come down if we need to, if efficacy is okay before that. But we want to do that, and then in other therapeutics and increasingly in oncology, due to the emergence of immunotherapies, which don't have that same monotonic increasing effect assumption necessarily, you may also want to find the optimal biologic dose, or OBD, and that's basically the dose which is giving the best response, typically based on something, based on some kind of usual biomarker or some other kind of proxy surrogate measure of efficacy, biologic effect, typically. Then in phase 2, this is usually the first stage at which we're going to put the use of therapeutic on the population of interest, the population who has a disease of interest, for example. And we want to establish that A, we have a signal that this drug works, and B, what dose or doses may be of interest to bring forward to the confirmatory trial where we're going to establish that efficacy and safety to a high degree. So, proof of concept, or often called phase 2A trials, are basically that, try and find a signal, and that's the most likely case. Like many drugs fail at phase 2A. Basically, lots of drugs looked decent in the lab, but don't succeed when you give it to the patient of interest, and you don't see any reduction in response rate. And then the dose selection, it can be a complicated process, and it's an area of great research interest at the moment in terms of finding those optimal doses. And of course, just to say, like phase 1 can bleed into phase 2. You can have phase 1 B, phase 2A type designs, and obviously phase 2 can bleed into phase 3. So the dose finding part that data can then be included in phase 3 if you have a seamless phase 2, phase 3 type design. But we'll talk here just as if these were separate as they would be traditionally. And then phase 3 uses your pivotal trial for regulatory approval, where we want to establish that the drug is both effective and safe, with the power analysis usually based on the efficacy endpoint. So we want to show that this drug has sufficient actual effect in actual patients based on gold standard endpoints, preferably like overall survival. In oncology, we want to show that the drug increases the percentage of people who survive after X number of years, for example. And we want to do that in a large randomized controlled trial, double blind, with the highest level of ethical standards, as it were. And so we want to have a very high probability of being able to find an effect if it exists, and also establish in a large cohort of people that you have a safe drug as well, of course. And of course, phase 3 trials, very highly regulated. This is basically where the regulator becomes much more involved. You'll have a very significant protocol. You'll have inclusion-exclusion criteria. You'll have randomization criteria. You'll have obviously, an entire system to ensure blinding. If you have an adaptive design, there's a whole set of additional logistical challenges associated with that. So this is really where the most of the focus is in terms of the statistical aspects, in terms of the design aspects, in terms of the difficult things. This is obviously the case where after phase 3 or after 2 phase 3 trials, that's when a regulator is then going to make a decision about whether the drug should be approved for actual patient usage in the real world. And then you have phase 4, which is a kind of our post-marketing surveillance stage, which is the drug has now been approved. It's now available for patients, assuming it succeeded during the clinical trial approval process. And in effect, we now want to establish things like long-term safety, rare side effects, for the sponsor to maybe be interested in, as I said, additional indications, subgroup performance in terms of efficacy. But as I said, safety is kind of what we're mostly talking about here today. And so, a phase 4 trial or a phase 4 study is kind of almost separate from the phase 4 concept. Like phase 4 could really include all post-marketing surveillance and all phase 4 trials that we'll talk about today. But I'm going to talk about here mostly today phase 4 clinical trials or phase 4 studies, which is where we want to do a study where we want to establish evidence for some hypothesis of interest, primarily that the drug is safe under one or more criterion, in terms of one or more potentially rare adverse events. So we're particularly interested in rare adverse events because, as I said, a phase 3 trial is going to be quite large. It should find most common rare adverse events. But ones that occur on like 1 in 10,000, 1 in 20,000, 1 in 100,000, that type of magnitude, may not be caught in a standard clinical trial, which might have like high hundreds or thousands of subjects. So, when we actually are doing post-marketing surveillance, as it's a standard requirement, we may have evidence that comes up which suggests, oh, an adverse event occurred here, like a death occurred or a serious cardiac event occurred, and we believe maybe there's a causal link. Now we may need to look into doing a study. I'll talk about, how a regulator makes that decision in a moment, but that's what we're kind of interested here. As I said, like many phase 4 trials traditionally, like especially prior to 2007, would have been like sponsor led. They would have been a sponsor just doing a kind of trial after the fact for efficacy purposes, and would have focused maybe on safety as kind of a side effect. But the ones we're going to focus on today are where a regulator requires it. And in effect, we have these real world patients, we have these signals coming in. We may have some additional context from how the clinical trial process gone, and in effect, we have ongoing surveillance occurring, but then something occurs which means that a phase 4 trial seems like it may be justified or maybe requested by the regulator, such as the FDA. And so the FDA has specific language for that I'm going to use from now on. Obviously, similar language would exist for the EMA and other regulators like the PMDA. But in the FDA's languages, there's kind of two types of phase 4 trial or phase 4 study that they would require. And they're a post-marketing requirement and a post-marketing commitment. And we're going to talk about mostly about the regulations and the guidance given for PMRs, because that's obviously the case where, that's a statutory requirement, and therefore they tend to be more thorough in terms of what they would and wouldn't consider to be, important or valuable or allowable for those types of scenarios. And ineffectively, a post-marketing requirement is where the FDA basically says the sponsor has to run a phase 4 trial or phase 4 study, and they have to establish that the drug is safe under some criterion that either all adverse events or some specific set of serious rare side effects or adverse events of interest. And there were traditionally a series of cases where these types of studies, PMRs, were required. Accelerated approvals, oftentimes accelerated approval was conditional on doing a phase 4 trial, and basically establishing that the overall adverse event rate was not unacceptable. So you basically would get approval, but you would have to do a phase 4 trial. Similarly, and then probably increasingly, accelerated approvals are also phase 3 trials are being run, and then accelerated approvals based on a surrogate endpoint, so like say progression-free survival in a trial time to event study, but the phase 3 trial continues after approval to then have an analysis on the gold standard OS analysis. And that might happen if you're doing, say, group sequential design, for example. But phase 4 trial, let's assume that the phase 3 trial has been shut down, and now we have to set up a new study or trial. Also, a deferred pediatric type trial was also another criterion that existed prior to 2007. So obviously, the pediatric population very commonly won't be included in the clinical trial, but a condition of approval will be that after the approval occurred, that you should do a separate study in the pediatric population. I think there's also similarly, if you have the animal study, approval based on animal study, you could be asked to do a phase 4 trial to kind of, just establish safety in human population as well, although that would obviously be quite rare. I suppose the big one that was added in the 2007 FDA Act, particularly we'll talk about the 50503 requirement from the FDA, and that was really made in response to a particular event that occurred, well, a number of events, but the most famous one was the withdrawal, the voluntary withdrawal of Vioxx from Merck, which effectively was a painkiller in the context of diseases like arthritis, which was found to have an unacceptable rate of cardiovascular events like stroke and cardiac arrest, obviously very severe adverse events we're talking about there. And this was something that was very controversial at the time and was a large inspiration for the 2007 Act, which added these additional powers for the FDA, where in effect they can now ask for a phase 4 trial in response to what they say themselves, quote unquote, new safety information. So some signal has become available from standard post-marketing surveillance or from some other source, which suggests that the drug should have an additional trial to establish that it is safe under one or more types of adverse events that are of interest. And I should say that the FDA does in the legislation have to establish that this investigating the adverse events of interest could not be done just through continuing standard post-marketing surveillance. So there is a burden of proof on them to show that a study is the appropriate way for them to do it. But assuming that boundary is met, that barrier is met, then they can ask for a PMR, phase 4 study or clinical trial, and the sponsor is required to do so at that point. I won't talk too much, but also there's PCRs, or post-marketing commitments, or PMRs. Should be PMC, sorry. A post-marketing commitment is basically where the regulator and the sponsor agree to a phase 4 trial, and the regulatory requirements there tend to be a bit more loose. But this is obviously something where, as an act of good faith, you would hope that the sponsor would react, positively to putting forward the extra information that the regulator asks, to ensure that the drug, doesn't end up being in the PMR type situation. So if we're talking here about these types of studies, like ones that are required, we're talking here about large studies. We're talking about like thousands, at least hundreds, likely thousands of subjects for multiple years, 3, 4, 5 years potentially, at the cost of tens to hundreds of millions of euro. And there's references provided for these kind of cost ranges here in the references section at the end. And really, what we talk about here, like, okay, what's the kind of, how many of these phase 4 trials lead to something happening? It's probably higher than this number I gave here, 3 to 4%, which is really the estimated total proportion of all drugs that end up being withdrawn. whether there's a phase 4 trial or not. I would imagine that phase 4 trials probably have a higher probability than this, since obviously there's a selection bias. If you had a phase 4 trial, there obviously was a safety signal came up. But if you want a general indication of how many drugs end up being withdrawn from the market completely, we're talking here about 3 to 4%. However, that's just to say that's not the only thing that can come out of a phase 4 trial. There could just be some change to the labeling if it's found that some additional side effects were found that they weren't in the initial approval, the labeling under the initial approval, and that's probably far more common. Like I think it's around 20 to 30% of drugs end up post-marketing end up having to make changes to their labeling or withdrawn, and the majority of that's just changes to the labeling. And obviously the hope is, as an important point to make is that nearly every therapeutic has some side effects, has some downside. It's about balancing those with the potential to help patients, whatever their condition is. So full withdrawal is kind of the nuclear option, isn't used too often comparatively, but if you're going into a phase 4 trial process, that's obviously, maybe the first step towards that. If something underlying is actually happening in terms of like serious side effects or some other concerning outcome. So in terms of what phase 4 trials look like, there's a wide variety of different types of study or trial that are out there. But I'd say that kind of 3 main ones, and the 2 first ones are really the ones that are important here, which are observational studies and randomized studies. But there may also be qualitative studies which are useful to give additional information there. So I'll mostly focus on these first 2, observational studies and randomized studies. So observational studies are obviously, you're taking the subjects, the patients getting the drugs as is effectively, they're getting prescribed a particular therapeutic, they exist out in the world, and then you're effectively doing an analysis on them. And you can do that either retrospectively or prospectively. As I mentioned before, the FDA, the EMA, other regulatory agencies will maintain their own databases of safety signals, safety outcomes, reported adverse events, like the FAORs is the FDA's big one for drugs and biologics. And in that case, you would just go to the you would go to the registry or to some other registry that you might do internally, or you might set up a specific registry for a study, and you basically just take, here's the 1,000 subjects or 1,000 patients who have got the drug of interest. We're going to follow them up for 3 years, and we're going to see what happens, basically. Or you can do that, 3 years after the fact, you might be asked to do that effectively by the FDA, and that'll be a retrospective study. Or you could do it prospectively, where you're now, you haven't got the data yet, and you're now going to set up a study to actually do so. Now, unlike a randomized trial, you're not going to randomize to one group or the other. You're just going to look at what happened. Take 10,000 subjects. You're going to see which one gets the new intervention, which one gets the standard intervention, and you're going to make a comparison based on that. But they will exist within a study system. So you might recruit hospitals, physicians, etc., to create that population for you that you're planning to do an analysis on, say, 3 years from now. And in terms of observational studies, there's 2 main choices. There's cohort-based analysis and there's case control type studies. So cohort analysis is very simple. You have a cohort of people who are getting the treatment. So that's just will emerge naturally after approval, you hope, obviously for a drug that's actually succeeding in the real world. And you either get one cohort, which is just your treatment cohort, the people who got the treatment of interest, the patients who got that of interest, and you might compare the adverse event rate for that compared to either some kind of standard amount of adverse events. So for a serious adverse event, a single serious adverse event like death may be sufficient for a drug to be pulled from the market completely. So you might just have we want less than X adverse events, or you might compare that to 1%. We expect like 5% of people to have an adverse event of one type or another. We want to ensure that the adverse event rate in our new drug is not higher than that in our cohort of people we're following who got the treatment of interest. Or you might have multiple arms in the sense that you might have a group of people at a given time who were prescribed your therapeutic, but who were another set who were prescribed the standard therapeutic at the time, or a basket of other therapeutics of interest. And you might then compare the adverse event rate between your new therapeutic group and the other group that didn't get the therapeutic of interest. So that's a cohort design. Case control is slightly different, where we basically get a cohort of people who had an adverse event of interest, and we have a cohort of people who didn't have an adverse event of interest, and we basically are comparing the proportion of each of those cohorts who had the intervention of the therapeutic of interest and who didn't. So obviously, logically, we're hoping that at least in the worst case scenario, that the adverse event rate, the proportion of people who had the therapeutic is roughly the same in the adverse event group and the non-adverse event group. And you may also match in other characteristics like age and gender, etc., of course, as well. if you want to do like a propensity score matching or whatever in the statistical analysis. But obviously, if in the adverse event group there's a greater proportion of that who are getting your therapeutic versus in the non-adverse event group, then that's obviously a signal of something of concern. Then kind of in between, it's really kind of in between this meta-analysis section between the two. But a meta-analysis, as you are probably aware, is where we take a large cohort of studies, randomized controlled trials, observational studies, other sources, and we basically, within all studies, typically there is adverse event rate reporting, and we basically take from all the estimates of the proportion of people who got an adverse event across all those studies, we can create a kind of a estimate which takes into account all the studies and also takes into account the study quality, etc. So obviously that would probably include the initial RCT used for approval, but it might include subsequent RCTs done comparing that therapeutic events to another therapeutic, or it could include observational studies, etc. And we create an improved estimate based on all of that data, and that's talked about in the FDA guidance for 50503. And I suppose just to say, a lot of what I'm talking about here is based on the literature. There's a lot of papers at the end of this, but also very strongly inspired by the FDA's own guidance for the specific requirements for phase 4 trials or for post-marketing surveillance in response to, as I said, the new safety information requirement, this 50503 requirement. But of course, if you wanted to go one step further, and I would say, as usual, the FDA would love there to be a comparison group, even in a cohort design, they'd rather there be 2 arms, not 1 arm. But they do realize, they say explicitly that, if it's justified, either due to practical considerations or just statistical considerations or whatever, then 1 arm trial is acceptable in a phase 4 trial, and it's very common, to be frank. Like most phase 4 trials I found during the research for this back in the day and for this webinar were 1 arm trials effectively. But you could do a randomized study. So you could do a randomized control trial. So of course that's basically like your phase 3 trial. You have inclusion exclusion criteria. You have tight control over the types of subjects. You have tight control over how the treatment is given, et cetera, et cetera. So you control the entire thing. You have a protocol, and you set out in clear terms what's going to happen in the treatment group, what's going to happen in the standard treatment group or the placebo group or the control group, and then you're going to randomize between those 2 groups. So that's a full-on RCT. However, an alternative proposal for phase 4, which is maybe more practical because it's significantly less onerous, is a large simple trial, which is where rather than having tight control over inclusion, exclusion, etc., what you effectively do is that you recruit a series of hospitals or physicians, etc., and you ask them to randomize each subject between like the standard treatment and the new treatment, the treatment of interest, the therapeutic of interest, and you otherwise don't interfere effectively. You just have standard clinical practice other than that. So, everything else is exactly the same, but so obviously all that inclusion, exclusion criteria, tight control is there. And the hope is that while obviously you lose some of the kind of control that you have in terms of the statistical analysis and the like everything kind of being statistically valid, you do get some generalizability there potentially that you wouldn't get from an RCT. And of course, as I say, a phase 4 trial is really where concerns like generalizability, where we're kind of looking at the real world, not just kind of the tightly controlled RCT world from phase 3, is of more interest at that point, right? And I suppose just to mention, there's lots of other studies which could potentially be of interest, which may or may not be sufficient on their own. That's obviously something that will be a negotiation between the regulator and sponsor. So you have animal studies, in vitro studies, pharmacokinetic studies, interaction studies, natural history studies, and probably more complementary studies like physician experience studies, drug utilization studies, etc. Those latter 2 kind of focusing in on kind of qualitative and quantitative information about how is the therapeutic actually being used in the real world, and how physicians feel about how it's gone. Okay, so that's hopefully a useful overview of phase 4 trials, and obviously post-marketing surveillance is a concept as well. So let's assume a phase 4 trial has come up. Let's assume it's related to something like a post-marketing requirement or post-marketing commitment. How do you now design? How do you choose the appropriate design, and how do you choose the appropriate sample size for that study or clinical trial? So obviously the first thing to keep in mind is the trial objective. Now, if this is a post-marketing requirement, if it's something the regulators ask you to do, safety is going to be what's of primary interest. And of course, then you have to ask the question of like which adverse events are of interest. That would probably be related to what the new safety information, how that was defined, or maybe related to, any safety event, adverse events that occurred during the phase 3 trial, if this is something for, like, say, the pediatric population or based on accelerated approval. How rare is the side effect? That's going to have a large influence on a bunch of other decisions further on. And obviously, if there's any subpopulation considerations, how's that going to affect things as well? So, for example, the FDA has a separate guidance they released last year. It's included in the references, talking about how phase 4 trials can be used to improve the diversity or representativeness of the trial. Basically, phase 3 trials often aren't fully representative of the expected patient population. A phase 4 trial as an addendum to all the other considerations, and the primary one being finding for safety. Maybe let's get a more diverse set of population to deal with that consideration as well. For efficacy, and this is probably more of interest to the sponsor than the regulator, you may be trying to just verify phase 2, phase 3 efficacy in the real world, quote unquote. You may be looking for certain subpopulations who may benefit more or less from your therapy or therapeutic. And of course, it said in additional indications may be of interest. We're going to talk mostly about safety because that's the kind of ones that are required. But most sponsors are doing some type of what you would call a phase 4 study all the time, because that's really useful information for improving how the drug is actually used by patients, by clinicians in the real world. So in terms of picking a design, like for a phase 4 trial, as I said, the most common is a cohort design. And for a very simple reason, it's the simplest, it's the easiest type to do, whether it be retrospective or prospective. So if it's retrospective, it's just, find all the people who have been prescribed your drug. Now let's just check how many adverse events you see in that cohort. Or, you may just be looking at the people who've, on the registry who have adverse events who have the drug of interest or the therapeutic of interest. So it's very easy to do a cohort design, whether retrospectively, which is to say based on existing data, or prospectively where you're actually going to recruit people. But there's causal issues that are of concern here. If it's a single cohort, if it's just a one-arm design, a cohort, no other comparator, you're going to have to ask yourself, well, what is the rate that we would consider to be unacceptable? Is that something we're getting from the regulator? Is that something we're getting versus the expected adverse event rate for the kind of what we expect from the wider population who are getting the standard treatment? Where do you get that from? Is of interest. And if you have multiple cohorts, as I say, the RCT is designed to kind of eliminate the kind of confounding effects, the collider effects, those kind of causal problems that if you're familiar with causal analysis, which are very problematic. A cohort design doesn't, or is much more difficult to kind of get rid of those completely. So you should be cognizant of those when talking about doing a cohort design. But it is considered mostly to be broadly acceptable for this purpose. For case control, obviously, this does add in, now you have matching. So from a causal perspective, it does, with some fairly reasonable assumptions, hopefully allow you to be more confident that the estimate of how much higher the adverse event rate is or isn't than the existing standard regimen is more justified in that case. But obviously you have to do the matching, you have to do a more complex statistical analysis like propensity score matching or something like that, especially if you're including multiple matching criteria, not just matching based on have adverse event versus not have adverse event. So more assumptions, more complexity there, but perhaps brings you a little bit closer to something like a randomized trial. A randomized trial, of course, the gold standard here, RCT, the proper gold standard, a large simple trial has many of the same advantages, where obviously, because randomization kind of removes the causal problem of everything else. Like if the only reason you got it is because you're randomized to it, then it's not because you were older or younger or sicker, or you had certain certain conditions or certain disease, symptoms, etc. So you're removing those kind of confounding collider type effects. So it's a much easier to make a direct comparison, because you're just saying, here's people who were randomized to get the standard treatment, here's people who got randomized to get the new therapy, the therapy of interest. Compare across them, and you have much more confidence in the statistical validity of any comparisons there. But they are more expensive, they're more difficult to run. They will probably, by definition, have to be prospective rather than retrospective, which itself is going to be more expensive. And of course, they could be potentially considered to be unethical, right? I find, that when talking about randomization to physicians or to patients, that randomization is something that people aren't necessarily all that comfortable with overall. So in the face of a phase 4 trial where the drug's already been approved, randomizing someone to not get the new drug, which obviously presumably was approved on the basis that it does something special or different or has particular effect, better effect or a better effect in certain subpopulations, could be considered to be unethical and considered problematic if you're trying to do it. So definitely something to keep in mind. And then you have your incidence rate effect. So thinking about the adverse event rate, how does that affect things? If we're going to make a comparison and say a cohort design, are we going to get that from a historical rate? Are we going to get that from previous clinical trials or a meta-analysis or other observational studies? If we have the standard treatment, like what is the definition of standard treatment? Like if there's multiple, the standard treatment could include people getting a bunch of different types of the drugs. They're all in the same class, but they're all different. Like, there's like for most medicines in common areas, there'll be like 4 or 5 or 6 different other medicines already being used for treatment in that areas, like different types of PPI or different types of painkiller. And so, the complexity of what the standard arm should be in a cohort design can be complex. And, do we need to split that into like several different standard arms or just have the kind of, here's the new therapy or the approved new therapy cohort, here's the other cohort. Is that sufficient to do the statistical analysis or the analysis you think you should? Considering what type of endpoint you have, like should you be treating it as an incidence rate, like a count type data, a proportion, a binary type data, like, is it like how many events people per person basically, or is it just, 50% of people get, 5% of people get the event, had an event in this group and then 5.5 had it in the other group? How are you going to deal with that? You could even do a survival analysis or continuous outcome. That's much rare, but count or proportion are the two common choices here. And then, of course, and I'll talk about it obviously in context of nQuery, how do I ensure that the study is appropriately sized or will be appropriately sized and the database is big enough, if you're using existing retrospective data, to be powered to do the hypothesis of interest. And if it's registry data or it's recruited data, that has a big effect on kind of, plausibly what kind of size of study I can look at. So if we're thinking about sample size determination, do certainly think of the objective of interest. If you're talking about a rare effect, logically the sample size requirements are going to be high, but also consider the practical problems, which is that, if you're doing a registry-based study, that's going to be a lot cheaper than a randomized study. And it's going to be a lot easier to run as well. But of course, do consider, as I said, the statistical, like, you get that simplicity, but it's the cost of perhaps more difficulty in having confidence in this, whatever the statistical validity of your actual results. Obviously, if we're talking here in the case of a phase 4 study that's been asked of you or required of you by a regulator, you do need to keep in mind the allowable design types, and obviously you'll probably have a negotiation about what the appropriate design would be. So keep in mind those kind of practical, considerations, not just kind of your more high-minded statistical considerations. The sample size method should obviously reflect those upstairs design decisions, like what the endpoint is, what the design is, etc. And obviously kind of take into account, like, well, what is the quote unquote acceptable adverse event rate? Is it literally one adverse event for a serious adverse event, like cardiac arrest? Is that too high where the drug could be, got rid of because of that? That's very different from saying, 1% of people have an adverse event, doesn't matter what it is. We don't want to be any higher than 1% type of situation. And like, where are we going to get that background adverse event estimate from for our sample size calculation? In terms of what our criteria is, it would typically be power, but you could also base it on the precision of your estimate. So, and just to be clear, if this is a phase 4 trial done in response to a regulator requirement or a regulator request, you're powering for the safety endpoint. That's the big difference between a phase 4 trial and a phase 3 trial. You're powering for the safety endpoint, not the efficacy endpoint. Now, you might be doing an efficacy analysis, and maybe you want to do a separate supplementary power analysis just internally in terms of communication with the sponsor, but the powering should be for the safety endpoint if we're talking especially for a post-marketing requirement study. And, we should think then, because, phase 4 trials are going, like, if you're looking at something like a rare event, large sample sizes may become quickly very impractical or very expensive. How could we improve the efficiency? And it may be the case that it's much easier to get controls than people on a new therapy, because a new therapeutic, because of course the new therapeutic is only just out there. It's obviously going to be more expensive. The control arm, it's much easier to get historical data. So it may make more sense to include lots more controls than lot more, like, for people for the new therapy, even though the overall sample size might be higher. because the cost per control is so much lower than for cost per treatment effectively. And if you're doing a Bayesian analysis, which is something which I think the FDA and other agencies are more open to for a phase 4 trial, it's one area where I think they've seen great promise for Bayesian analyses historically, then integrating historical data might be able to do that actually directly into your analysis using something like a power prior. And if you're doing multiple adverse events and you're kind of, each adverse event is itself a decision criteria, then you may want to consider something like a Bonferroni adjustment, like where you divide the total alpha, like 0.05, by the number of analysis. So, 0.05 divided by 5, if you have 5 separate adverse events that you want to do a physical, like a kind of a hypothesis about each of them, basically, that would be sufficient to reject the drug. And you can see here on the right, basically some sample size formula from the McKean-Campbell book, sample size for clinical studies. It has a slightly different name now. I've included both the one I've used, which is the second edition, which has that name, and the subsequent third edition, which is a slightly different name in the references. But the practical information is effectively the same. There's some changes in language here, but the top two there are basically for a single cohort design, where either you don't have a background incidence event, you basically just want to see one or more events or two or more events would be considered to be unacceptable, where the drug would have to be withdrawn or there would be severe consequences. And we just want to ensure, we have enough people to ensure that we can say with confidence that we won't have, like, given some underlying rate, that we won't get more than that effectively. And then we have known background incidence rate, which is where we have like a rate that we assume is the historical rate or the standard treatment rate, like 1%, and we want to compare, like we want to test our rate versus that 1% rate under the null hypothesis. Unknown background is basically a 2-arm cohort design, or basically the same for a 2-arm or CT as well, or a large simple trial. But basically we have 1% in group 1 in the control, in the standard treatment group, and then we just want to reject the null. We don't want to reject the null hypothesis that that they're the same, basically. All right. And so you do an unknown background analysis there. And then we have a case control study, which we talked about previously. So, these are kind of the formula you might see if you go to the McKean book, which has a separate chapter on the post-marketing surveillance, they call it. But basically, these are for phase 4 studies. So we don't have too much time available, so I'm very going to quickly go through these examples. And I've got a couple of real-world examples of sample size determinations used to justify some post-marketing studies, some phase 4 studies. And so in this case, we had insulin degludec, basically assessing its safety. And in this case, it was any adverse event. And so in effect, the kind of sample size calculation is a bit weird, but effectively what they were arguing here is that they wanted to have sufficient sample size to be able to find at least one adverse event for given any of those adverse events might have a rate of greater than 0.1%. And actually, if you include all adverse events, the overall any adverse event was like 20% or something like that in this study, if I recall correctly. But this is it's almost a little bit weird. It's basically it's really a power calculation for all the adverse events that could potentially exist, since this was a post-marketing surveillance study interested in all adverse events, including things like hypoglycemic events, injection related events, allergic events, cardiovascular events. There was separate sub analyses for severe adverse events, but the power was based on just any adverse event. And so you can see here that they thought they would need 3,000 subjects to basically, let's assume there's just one adverse event of interest, that they would need 3,000 people for an adverse event that had a 3-year incidence rate of around 0.1%, and they wanted to know, to have at least, to be able to find at least one of those if it actually existed. And they wanted that with 95% probability, which is basically our statistical power. And you can see there was a dropout there rate of 4,000. So if you had 25% dropout, you end up with 4,000 subjects. So you can see the expected incidence rate here was 0.0001. So, it's obviously 0.1%. Divide that by 100 to get that on the proportion scale. The number of occurrences is 1. So we get that there. You will note here that it does mention here briefly that, if you're interested in the case where, no adverse events have occurred, how could you do a sample size calculation there? Well, you could basically go, well, what's the upper limit for the adverse event rate, given that 0 events have happened? And you can basically do a sample size calculation based on that as per the suggestions here. And you can see it's like the n would equal 3 divided by the expected incidence rate for a 95% confidence interval. You can look at McKean and Campbell, they cover that more in the book. And then we have a power of 95%, and this gives 2996, which is obviously slightly smaller than 3000, but for practical purposes is effectively exactly the same. So if we put in 3000, you'll see, slightly above 95% power. And you can see here in the output statement, in a cohort study with no background incidents, the probability that one or more adverse event reactions will occur in a sample of 3000 with anticipated rate of 0.01 is 0.05. This means the power is 95%. Okay, so obviously we don't want any adverse events to happen in this particular study in their particular sample size calculation. And if you look at sample size calculations for post-marketing studies that are available, most of them will look something like this, and they'll often reference the McKean table, where he provides, the sample size required for a wide variety of different values of A, OR, and power. Okay. So in terms of, say, if you were doing a different type of cohort design, it's a two-arm cohort design. Sorry, no, this is a one-arm cohort design. But basically, this is one in which there's an underlying incidence rate, and effectively what you want to do is to establish that the incidence rate is no higher than 0.9% effectively. And you effectively go, okay, let's just say it's actually 1.8 or it's higher 1.8. What's the chance? What's the power we could reject that effectively? So if we go to the other post-marketing surveillance table, POT15, and we enter 0.05 at the 2% level, the background incidence rate was 0.009, and they give in that sample size determination, like a double is what they say the new treatment could be maximally allowed to be. And what they do then is that means the additional incidence rate, which is kind of the how much higher is it than the other one? So basically, the higher one minus the baseline rate is 0.09. I give the kind of calculation here in the table on the right here, if you're interested. And we then set the number of patients to 1200. And you can see that we get a power that's a bit above 80% here. So what you'll often find with these calculations, and even if you go into the chapter in McKean's book, is that they suggest rounding up to the nearest 100 or the nearest 1000. So if I actually do the calculation exactly, with the power to the 80%, it'll end up being closer to 1100. So 100 less than what they required, but that could be seen as them rounding up, or it could also be them adjusting for dropouts. So for example, with this sample size, you'd have an allowance with samples of 1200, you'd have an allowance of dropout of 8%, and you would still have sufficient sample size to be powered appropriately to be able to find this additional incidence rate if it existed. I suppose it might be useful to contrast that with a two-arm trial. Effectively, what would be the equivalent sample size? So, this is a two-arm trial, two-arm cohort design, I should say. And, the inputs to this are very, very similar to what you saw in the previous example. Let's say just set the controls per case to 1. So for every control, there was a case. So, basically the size of the therapeutic cohort and the new therapy cohort and the standard treatment is the same in this case, and we set the power to 80%. The number of patients here is 2032. Sorry, this should be two-sided. Apologies. Yeah, it should be 2580. And you can see that that's actually just the number of patients required in the treatment group, in the group that got the therapy of interest. And so you can see that's already twice the original one, but you actually need to double this again for the total sample size. You're going to include the control cohort as well. So obviously, that's a 4-time increase. But one interesting thing to note here is that, of course, if I was to repeat this calculation, but to so if I copy and paste this calculation across, but I change the number of controls per case, let's set that to 5. So for every Every subject who got the new therapy, we have 5 controls. Let's say controls are much easier to get via, going from a registry or something like that. And we set the power to 80%. The required number of treatment subjects is reduced significantly from 2580 to 166, so nearly a reduction of 1,000. Though note the overall sample size is now equal to that plus from the control group 5 times that. So obviously that overall is a fairly significant increase. So it's just something to be aware of there. So if you take this, if you did multiply this by 6, then that would give you the total sample size required, which would be much higher. But of course, you have far more controls there, which may be substantially cheaper by a magnitude. So the total sample size here is actually 10,000 here. So that's nearly twice the sample size. But if you're looking at a retrospective registry, getting that might be those subjects probably already exist, right? Because there's probably a lot of people who've had the condition of interest over a long time over that period. And then very briefly at the end here, just a case control study. I didn't find any from the literature in this case. I'm just going to take this directly from the book I've referenced here many times and from which these formulas here are taken. And basically, this is a case control study, and you can see the summary here and then what was input to nQuery here. But in this case, effectively, the inputs are pretty much exactly the same as what we saw for a 2-arm cohort design, but obviously the sample size requirements will be quite different. So we have a 0.05 at the 2-sided level, additional incidence rate of 0.01 and 0.05. So the 5% background incidence rate, a 1% increase in the incidence rate for that we're trying to ensure we're not above. For let's say one control per case and a power of 80%, we'll get 7277. So this is actually a one-side analysis, apologies. So 7227, so about 14,000 basically in this case. And if we were to repeat this again by increasing the number of controls to say 5, once again, a very substantial reduction here from about 7,000 to 4,000, so maybe like 40% or so. Then we've significantly reduced the number of subjects we need from our new therapy group, in that case. Oh, sorry, we've significantly reduced the number of people we need from the adverse event group. So we only need 4,000 people to the adverse event group, but we do need a lot more people who have not had an adverse event. So in this case, we need 7,000 people who had the adverse event and 7,000 people who haven't had the adverse event. In this case, we only need 4,000 people who had the adverse event, but we need a lot more people who didn't have the adverse event. So, for example, the total sample size here would be around 14,000, but the total sample size here would be 6 times this, which would be around 27,000. So overall sample size much higher, but we need a much smaller amount of people who have had the adverse event of interest or adverse events of interest compared to this design. So obviously, that's a trade-off you have to make based on the relative cost or difficulty of finding people with the adverse event versus not having the adverse event. Okay. So I think we're basically at the end of the time there. So hopefully those examples give you kind of a brief flavor of what's an nQuery for these. I would say that most of these methods are effectively just some equivalent of an inequality test for one proportion or an inequality test for one Poisson rate, and those are available as separate tables. But these post-marketing tables are designed to mirror the kind of methods as they're described in the McKean and Campbell book, but which is also referenced it also references a number of papers which they take those formula from. So in terms of discussion conclusions, post-marketing surveillance monitors therapeutic safety after approval, after the drug is available to real-world patients, with a particular emphasis on rare side effects, on rare adverse events. Phase 4 studies may be required or may be asked to evaluate the safety in cases such as accelerated approval or where new safety information emerges. Also, phase 4 studies may be done by a sponsor separately to check efficacy and for safety maybe as a secondary endpoint. But today we're focused mostly on those ones that are where a regulator has asked or required a phase 4 trial or study. Phase 4 studies have a wide variety of options compared to, say, a phase 3 design, with observational designs, cohort or case control type designs being very common and for very practical reasons, effectively, especially if you're using a registry-based study, for example. And many phase 4 studies, despite this, are still undersized for the hypothesis that they're supposed to test. especially if we're talking about detecting rare adverse events. And so sample size determination methods like those described in the McKean and Campbell book and which are available in nQuery may be useful to do an evaluation to find the appropriate sample size for your study. So that concludes today's webinar. I want to thank you all for coming today. Just a couple of small things if you're interested. If you want to try nQuery, either because you don't have nQuery right now or because you have the base tier and you want to try the pro tier or expert tier, you can try nQuery with all of its tiers available at statsolves.com/trial, and you get a 14-day free trial. No downloads required. It's basically, you can do it all within your browser, and you can use that to get a flavor for if nQuery has the options that you think are appropriate for your clinical trial design problems. And if you want to get in contact with us, you can email at info@statsolves.com. If there's a few questions here. I'm not going to have time to get them today, unfortunately, but I will be answering them all by email. Don't worry about that. But if you want to get in contact with us after the webinar, you can email us at info@statsolves.com. And of course, that email and all the other information will be sent with the slides, which will be sent alongside the recording later today or early tomorrow. And if you want to know more about nQuery generally, you can visit our company's website, statsolves.com, where you'll find the archive of previous webinars and worked examples and more information on all the tiers of the product. As I mentioned, there's references available at the end of this webinar covering the kind of regulatory requirements I've referenced quite frequently during this webinar, some general background on post-marketing phase 4 trials, some phase 4 trial summary in terms of average cost, average failure rates, etc. Kind of meta-analysis is there. And then the specific design and sample size requirements. So without any further ado, I just want to thank you so much for attending today's webinar. I hope you've learned something useful about phase 4 trials and post-marketing surveillance and the requirements that may come up in that space. Our next webinar will be coming out in August, which will likely be about uncertainty in effect size estimates and how that affects sample size and power calculations. And that will be presented by my colleague, Calvin O'Brien. So I want to say thank you so much for attending today's webinar. I hope you have a great day. And as I said, the webinar recording and slides will be sent to you later on. So if you need to review or peruse further, hopefully that will help you with that. So thank you so much. I hope you have a great day. Thank you so much and goodbye.